Okay, so welcome back to my channel, and in this video I will be explaining how you can analyze the raw sequence data of 16S rRNA partial gene sequence, which has been done with the help of Sanger sequencing method. So generally when you provide your samples for 16S rRNA gene sequence analysis, you will get two different kinds of the sequences for the same organism. So one of this is the file which is obtained from the forward primer, and the another file that you will get is the file that has been obtained by the reverse primer. Okay, so you have to first of all trim these two files, and then you have to merge these two files to create a continuous contig. Okay, but before we begin, I will first of all show you what is 16S rRNA gene and how this sequencing basically works and what kind of data you will get. Okay, so suppose if this is your 16S rRNA gene, in order to identify the sequence of this gene, what basically is done is that once you purify the DNA and you provide the specific primers, so you provide a specific primer over here, and so this is basically called as a forward primer, and you provide another primer which will bind over here. This will be basically called as a reverse primer. Okay, so this entire 16S rRNA gene is around 1600 base pairs. Okay, but now what we are doing is we are doing its partial sequence analysis, so we are not identifying the entire 1600 base pairs, but we will be identifying only a specific region or the partial region of the 16S rRNA gene. So in this case, basically what will happen is that the forward primer will start reading the sequence from this end, and it will go into this direction like this. Okay, so this is how your forward primer will go. And so for the forward primer, what will happen is that you will get a sequence which is been read from left to right. Okay, and it will be written in this direction. Like suppose if this is here, if there is A T G C, then it will be written in the form of like A T G C, in this way. Okay, until the sequence continues. But in case of the reverse primer, what will happen is that the reverse primer will start reading the sequence from the opposite end, and it will keep on reading the sequence like this. Okay, so the first file you will get will be the file of the forward primer, and the another file you will get will be the file of the reverse primer. So I will just show you these two files. So if you just see onto my desktop, there are two files. One of the file is of the sequence coming from the forward primer, and another is the file which is coming from the reverse primer. Okay, so now basically what we need to do is that we need to combine these two sequences in such a way that we get at least this much amount of sequence with us. Okay, now dealing with the forward primer is very easy, I will show you that, but dealing with the reverse primer is slightly tricky. Why? Because the sequence that is been mentioned, or the sequence that will be given to you will be in the reverse manner. So suppose if there is A T G C G C C. So if the sequence is going like this in this direction, your sequence file will be starting and it will read in this direction. Okay, so if the sequence is like A T G C G C C, then your sequence file will read the sequence as A T G C G C C. Okay, so it is actually the reverse order in which you will be getting your sequence. Secondly, these two sequences that you will get will be complementary to each other. Okay, so what you need to do is that you will first of all need to reverse the sequence, and then you need to create its complement. Okay, so if you create the complement of the reverse sequence, then you will get the sequence which is like this. Okay. Okay, so if this, let me just show you with the another color. So suppose if this is the original sequence, okay, which has been read in this direction, then you have to do its reverse complement to get a sequence which will start from here up to here. Okay, now once you do both these steps, what we need to do is that we have two sequences, which will be one like this, which is of the forward primer, and the another which is again the reverse complement of the reverse primer, which will be again something like this. Okay, now we have to merge these two files or these two sequences and create a continuous sequences or a continuous sequence, which is basically called as a contig. So this is the entire process regarding how to analyze the 16S rRNA gene, and I will explain you step by step how you can do it. Right, so let me just go back to my computer screen. So first thing that you need to have is the reference files of the forward primer and the reverse primer. So if you have a forward primer, somewhere it will be written as F. So let me just zoom. So if you just see, it is F over here, and the file name will be the same, but there will be R over here. Okay, so F and R, so both these files belong to the same DNA sequence, but one is from the forward primer and another is from the reverse primer. Now to analyze this, we need to have a software which is called as a bioedit, and this software, which is a bioedit, it is a free software, and you can easily download it from the Google, or you can simply Google the software with the name bioedit, and it will take you to the page from where you can download it. It is a very small software and it is very easy to install, so you simply install it onto your Windows system. So once you have done this much, what you do is you open the software, which is this bioedit sequence alignment editor. So once you have opened this, we have to open any one file first. So first of all, we will start with the forward primer. So what we will do is that we will go to files, then we will go to open, and then onto the desktop. Here I'm not finding. Okay, so onto my desktop, you just search for the forward primer and open. Okay, so now when you open your file over here, what will you see is that two different windows will open. So I will just drag this window over here. I will increase the size of it, and I will also open this window and I will do like this. So now if you just see, this is basically a chromatogram of the sequence that you get. Okay, and this is the sequence. So if you just see this sequence, and the sequence which is mentioned on the top is one and the same. Now these are the two windows, and we first, what we need to do is we need to focus on the chromatogram, right? And you just focus on the chromatogram, and here if you carefully see, one peak is one nucleotide. So wherever you find a single peak, so all the peaks of G for a nucleotide G is of this black color, then all the T's will be of red color, and then you have A's which is of green color. Okay, so now you need to understand this, that at any given location, there has to be only a single peak, not a multiple peak. So for instance, if you just see the original, not the original, the initial part of the sequence up to up to say 11, 12, 13, 14, 15, 6, 7, 18, up to 17 nucleotides. If you just see, there are the sequence, if you see, multiple peaks are coinciding at a same location. That means this part of the sequence is not good. So what you need to do is you need to trim this down. Similarly, you need to trim the sequence from the terminal part as well. So if you just see over here, after say around 400, okay, so this region is not appropriate, so this much you first need to trim. Okay, so now always remember, when you are trying to trim the sequence, you have to first trim the end part of the sequence, not the initial part of the sequence. So let us just go to the end part. So we are at the end over here in the chromatogram, but on this window we need to go to this end part of this sequence, which is over here. And now if we just carefully see, we don't want the sequences, say probably from this location. Okay, so we don't need this region. So directly we cannot delete it from here. What we need to do is we need to play around with the sequence which is there in the window above, so this one. Okay, so first thing you do is you go to this place onto the this window where it says mode, and you change the mode to edit. Okay, you so first go to this mode and change it to edit. Now select the nucleotide from where you want to trim. So for instance, let me just, for my safe side, what I will do is I will just start trimming it from this nucleotide. So from after 390, so this nucleotide I can go up to G, so I will go up to G. So the sequence quality is good, so I will go up to G and I want to remove the terminal part. Okay, so after whatever is there after this G I want to delete. So for that, what you need to do is, first of all, you need to keep your cursor which is blinking just below the nucleotide you want to delete, like this. And then you go to this option on to the taskbar, or on this you just go and search for this option select to the end. Again, you go to this edit tab and then you select to the end. So as soon as you do it, your sequence will be selected. Once you select the sequence, what you need to do is, again you go to this edit button and then you simply click on delete, so that this part is deleted. Same thing you now need to do with the initial part of the sequence. Okay, so if you just see the initial part of the sequence, we can say that anything which is after 20 is good. So we will keep our cursor floating around at the 20th nucleotide. Now you just go to this edit button and then you just select this option select to the beginning. So what it will do is it will select whatever is there after this G or before this G from the beginning. Okay, and then you can simply go to edit and delete it. So we have deleted the initial part and the terminal part. Okay, so depending on the chromatogram. So chromatogram is very important, right? Now what you do is you have to copy this sequence and you have to paste it somewhere in your notepad file. So what we will do is we keep our cursor at the first nucleotide over here, and then you go to edit option and then you just go to select to the end. So from the first nucleotide to the end of the sequence, it will copy your sequence. So it will select your sequence first of all, and then you go to edit button and press on copy. So you have copied the sequence. We will go to our desktop and we will create a new notepad file, simple text document over here. I am just keeping it over here. You can open it and you can save this file into a faster format, like we will just say F for forward, and then you can paste the sequence and we will simply save this. Okay, so we have trimmed the forward sequence, forward primer sequence, and have pasted this sequence into the text editor. Okay, so this is what we need to do with the forward primer sequence. Now we have, what we will do is we will open in the same way the reverse primer sequence. So again, as I have mentioned earlier, you will get two different windows like this. I will do this time quickly so that you can get the understanding of this better. So first of all, you change the mode to edit. First you go to the end of the sequence. Even over here we will go to the end of the sequence and we will trim the sequence from 390. So whatever is there after 390 we will delete it, because if you just see, this quality of the sequence is not good. Okay, so we will try to trim this part. So we'll go to, you keep your cursor at the place after which you want to delete the entire sequence. So the cursor, I have kept it just beside A. Then you just go to edit. Okay, I will just go to this A. I'll go to edit and select to the end. So from this A, whatever is there on the right hand side up to the end, it will select, and then we will go to edit and simply delete it. Similarly, we will go to the front of the sequence over here and even over here. Okay, and we will delete first 20 nucleotides. So what we will do is we will delete first 20 nucleotides. So I will press keep my cursor at the 20th nucleotide. I will go to edit, select to the beginning, so it will select everything what is there up to the beginning part of the sequence, and then we can simply delete it. So now we have this reverse primer which is trimmed sequence. The next thing that we need to do is to create the complement reverse of it, or reverse complement of this. So for that, what you do is you select the sequence in this window, the window which is on the top, and you select the sequence on this left hand side panel. So once you select this, okay, so it will turn into a black color. So if you just, I will just click on this, it has turned into black color. Now what you do is you just go to sequence, go to nucleic acids, and then there is an option which says reverse complement. When I press on this button, you just focus on the sequence which is there on the upper window. So now I'm clicking on it and just keep an eye on the sequence which is there on the upper window. So as soon as I click over it, the software has converted the reverse complement of the sequence. Okay, so here what we see on our screen is a reverse complement. Okay, so we need to copy this sequence. So you just keep your cursor over here at the beginning of the sequence. Here what we are doing, we are simply trying to copy the reverse complement of the reverse sequence, so reverse primer sequence. So you just keep your cursor at the first nucleotide, go to edit option, and then you go to select to the end. So it has selected the entire sequence up to the end. Then you go to edit button and press on copy. So it has copied the reverse complement trimmed reverse primer sequence. That is what it is. Yeah. And onto your text editor, you give a name like this, R for reverse, and reverse complement like this, and you paste it. Okay, so what I have done, I have just saved one file into the FASTA format of the forward primer and the reverse complement of the reverse primer. So this is the two files that I have, okay, into the notepad. Now what I will do is that I will save both these files together into the FASTA format. So for that, you go to this files button on to your text editor, and then you just go to save as. Now you just go on to your desktop or any place where you want to save this file. Okay, you change the save as type to all files, and then you change the name as sequence dot fasta. Okay, as soon as we press on save, what will happen, the sequence dot fasta file has now been created onto our desktop. We will close both these windows for now. Okay, so the next thing that we will do is that we will combine these two sequences to create a continuous contig. Okay, so for that, what we will do is that we will open the FASTA file that we have saved. So we'll just go to open, go to desktop, and here you have to select the FASTA option, and then you open this sequence.fasta. So now if you just see over here, both the sequences together has opened onto your screen. Okay, now what you do is you first, again you just go and change the mode to edit as we have done previously. Press Ctrl button and select both the sequences. Okay, so control button and then click on the first sequence, keep the control button pressed and then click on the second sequence. So you have selected both the sequences. Then you just go to this option which says accessory application. Okay, and then you choose this which says cluster W multiple sequence alignment. Okay, don't change anything over here, keep everything, all the features as the default settings, keep the default settings, and then you just press run cluster W, give OK. Now a new window will open in front of you like this. Okay, so this is the new window that is there. You can minimize the older window. So now in this new window, if you just see carefully, on aligning both the sequences, okay, you are getting the forward primer which is going like this, and then the reverse primer, reverse complement, which is coming like this. Okay, so if you just see, they are perfectly aligned like this. Okay, so if I just show you a diagram, what we have got is that we have got something which is this. We have got these two sequence that are aligned. So this part is done. Now what we need to do is we need to create a contig. Means we have to merge these two sequence and create a single sequence. Okay, so for that what we will do is that we will go to this window. Sorry, yes, we will come back, we have come back to this window of the sequence, of the aligned sequences. So here we have both these aligned sequences. You just select both of these together, and then you go to this tab which says alignment, and here if you just see, there will be an option which says create a consensus sequence. Okay, so as soon as I click over it, what will happen, it will merge both these two sequences of the forward and the reverse, and it will then give me a continuous sequence. Okay, so I will get a continuous sequence over here. Change the mode to edit, and then we need to copy the sequence. So for that, to copy the consensus sequences, what you do is you just go and keep your cursor at the first nucleotide, then go to edit, and then you choose this option which says select to the end. So you have copied the entire sequence, select to the end. Okay, then you just go to edit button and then go to copy. Okay, minimize everything which is over there, open your faster sequence or the notepad, whatever is convenient to you. Okay, so I will just delete this and I will create a new file which says consensus sequence, and I will paste the sequence. Okay, so I have created the consensus sequence by merging the two forward and the reverse primer together, and we have the sequence with us. And this is of course a partial 16S rRNA gene sequence. We will copy it and we will do its blast to understand how well the sequence is. Okay, so for that you open the web browser and then you go to NCBI blast, and onto this NCBI blast you just go to this option which says nucleotide blast, simply paste your sequence over here. Okay, and do not change anything for now, and simply click on this button of blast. We will wait for few seconds till the results appear in front of us. Okay, so now the results of BLAST is there in front of you, and if you can see this, the top hits are from the Wolbachia species, 16S rRNA gene partial sequence. And if you just see onto this page, the first thing that you need to look in for is the query coverage. Okay, so your entire sequence should be covered in the BLAST results or BLAST analysis. If your entire sequence is not covered, if it comes say around 90% or 95%, then the terminal parts of the sequence you have not cropped it well. Okay, so when you are trimming your sequences, your forward primer and the reverse primer, if you have not trimmed it well, or if the quality of the sequence provided to you is not good and you are trimming it not in a correct way, then despite having the percent identity being high, make sure that your focus should be first on the query coverage. Okay, so you go back again, trim your sequences, make sure that first you reach to the point where your query coverage is 100%, and once you reach to this point query coverage, then you look in for percent identity. Okay, your query coverage should be 100% when you are say uploading your sequence to the gene bank database. If it is 100% and then the percent identity is say around say 50%, sorry, 95% or 97% or 98%, it is okay, that is not an issue. But whenever you are uploading your sequence, make sure that the query coverage is 100%. Percent identity, after you have adjusted your query coverage to 100%, percent identity can be less than 100%, that is okay. Depending on the type of the strain you have, depending on the novelty of the strain, your percent identity can be less than 100, that is absolutely fine. But if your query coverage is not coming to 100, it means that you have not trimmed your sequence properly. So do not directly focus on the percent identity before checking out the query coverage. Okay, so this is how you have to analyze your sequence. You can go to the graphical summary and the alignments. Okay, so this is the sequence that we had got. Okay, so if you just see, our query is 1, so our sequence will be always shown as a query, it is starting from 1. Make sure that whenever you are doing the same thing with your sequence, it should start from 1. If it is not starting from 1, again you have not trimmed your sequence properly. Okay, if it is starting from 15 or 20 or 15, it should not happen, it should start from 1. Your query should always start from 1. Okay, so this was all I wanted to explain about how to create the consensus sequences and how to trim and analyze and merge the forward and reverse primer sequences. Okay, I hope this video is helpful to you. I will catch you in the next one, where I will be explaining how you can upload the sequences to the gene bank database of 16S rRNA gene sequences. Okay, so thank you.